Hello, everyone, and welcome to the second episode of Psyche Sci on the Fly. We got awesome feedback from the first one, so we thought, why not go for a second? Today, we're going to be tackling a topic many psych students have difficulty with, which is why it feels like it comes up so often: action potential. And so with us to help us manage such a complex topic, we have Dr. David Ragsdale, an associate member of the Department of Physiology, focusing on improving the teaching and learning of neuroscience at McGill. He currently teaches courses to undergraduate, graduate, and medical students, myself included. David, thank you very much for giving us a hand with this.
You're welcome.
All right, first off, I think we should start explaining how a neuron is structured, just to put you all in perspective. So neurons actually have multiple different morphologies, but the most common picture of a neuron starts off with the dendrites on one end, which can feed into a cell body known as the soma. And from there, the axon, the long piece, carries the signal down to the terminal buttons, where it will carry the signal onto other target neurons or organs. And this process is instrumental for a number of various different functions within our body, such as movement, sensory information, cognition, and the list goes on. But the question here remains, how does this happen? How does a stimulus from pricking your finger on a nail, for example, find its way all the way up to the brain to be perceived? The answer is action potential. Dr. Ragsdale, I was just wondering if you could tell us the steps that go into this process.
Sure. All right. So I will give it my best shot. And I'll start by saying that I think a lot of your students are in psychology, right? If I understand this correctly.
Correct.
Right?
Correct. Yeah.
If you're a psychologist, if you are a neurologist, if you are a neuroscientist of most kinds, in the grand scheme of things, you're not actually, this isn't really very important, right? What you really need to understand is what action potentials do and what they're for. And so going forward in your life, even if your career is neurology or neurosurgery or something like that, it's important that you understand that the brain sends action potentials, but the details of the physiology are really only important if you're a neurophysiologist who studies action. Having said that, if you're taking a class where you're supposed to know this, well, you're going to have to learn it, right?
You're going to have to, yeah. And that's a lot of the people within the psychology faculty, for sure.
A lot of people in your psychology major are going to have to learn action potentials work. And then 5 years from now, all you're going to need to remember is that action potentials are these spikes that propagate information. But you all know that the outside of every neuron and the inside of the neuron are bathed by physiological solutions. And those solutions contain positively and negatively charged ions, right? And of course, that's why these solutions can conduct electricity, because they have charged ions, and it's the charged ions moving around that actually can conduct electrical currents. Neurons invest a huge amount of energy constantly pumping sodium ions out of the neuron and pumping potassium ions back into the neuron. So those concentration gradients and the membrane potential that they create, that's a source of potential energy. And the neurons use that potential energy to send information. That's the work that's done by the nervous system, is to send information. So that's why they put so much energy into pumping the sodium out and pumping the potassium back in. The impulses that we're talking about are propagated down long, thin processes called axons. And again, I'm 6 feet tall. And so there are axons that start in my toe and they end in my brainstem. So those axons are 5 feet long. So when someone touches on me on the toe, you
feel it almost instantly.
It's got to get all the way up to my brainstem, and so it propagates using these action potentials. So I'll describe the detailed physiology of that first. The axon, that long, thin axon, is completely covered with two kinds of ion channels. And one type is called the voltage-gated sodium channel, and the other is called the voltage-gated potassium channel. So voltage-gated ion channels are proteins they really do look just like bagels or donuts. So people can take very high-resolution pictures of these things, and that's what they look like. They're proteins that sit in the membrane, and they make a hole. There's a hole in the center of the protein, and that hole is selective for one specific kind of ion. So there are sodium channels that have a hole that only lets sodium ions go through. There are potassium channels that have a hole that only lets potassium ions go through. And there's lots of other kinds of ion channels too. There are ion channels for chloride, there are ion channels for calcium, and so forth. The other thing about ion channels is that they have gates, and the gates can open and close. So the port of the ion channel isn't always open. Now, in the case of voltage-gated sodium channels and voltage-gated potassium channels, when the neuron is just sitting around doing nothing, the gates are closed. And that means that The sodium on the outside wants to come in because there's a lot of sodium on the outside, but not much on the inside, but it can't come in because there's no way for it to get in.
There's no door that's open. Yeah.
It's closed. The same, there's a lot of potassium on the inside and not much on the outside. So the potassium wants to flow out, but it can't because those are closed as well. Action potential begins at the place in the neuron where the axon is connected to the cell body. So it begins there because the initial segment gets depolarized. That is, it gets more positive than its resting level. So typically it has to get about 20 millivolts more positive than the resting level. So say from minus 70 to minus 50. So what happens when the initial segment gets to minus 50? Well, what happens is the voltage-gated sodium channels begin to open. That's makes it the threshold. That makes it the threshold is that causes enough of a depolarization that the sodium channels start to open. And the reason that the voltage-gated sodium channels open is that in the channel, there is a sensor that senses voltage. It can sense when the membrane potential, when the voltage across the membrane changes. And so it senses that change, and that causes the channel to open. And this is all about
is there a name for the sensor, or is it just the sensor?
It's called the voltage sensor.
The voltage sensor, okay.
Exactly what it's called. The protein has some charged amino acids that are actually in the membrane, and they feel when the membrane potential changes, and they move in response to the membrane potential changing, and that's what opens up the ion channels. What happens at the threshold is you get some critical number of sodium channels to open. It might be, let's say, 5%. So let's say 5% of the channels open at minus 50. So what happens? Well, when those channels open, sodium ions start flowing in through those open channels, right? Sodium ions are positively charged. So when they flow into the initial segment, that makes the initial segment get to maybe minus 45. Well, at minus 45, maybe you get 10% of the channels open. So now you have even more channels opening, so you get even more sodium flowing in. So now maybe that causes the membrane to get even more positive, let's say to minus 30. Well, at minus 30, maybe you get 30% of the channels open. So now you got even more channels open, you got even more sodium flowing in. So now you get the membrane goes to like minus 10. Well, now maybe at minus 10, you get 100% of the channels open. So it's like a positive feedback loop, right? It's something that once it feeds on itself and drives itself forward. Because this is positive feedback, it always drives itself to completion. In the end, all the channels are open. And what happens is at that point is the membrane will depolarize to a fairly positive level. So the membrane creeps up to the threshold and then it spikes very rapidly up to around plus 30. Well, what happens next? Well, two things happen next. The first thing is that the voltage-gated sodium channels inactivate. So that means that they just shut off, even though they open when the membrane gets more positive, but once they open, they almost immediately shut. And they convert into a state called the inactivated state, and then they just don't open again. They won't open again. So that shuts off that inward flux of sodium ions. And the second thing that happens is the voltage-gated potassium channels open, because they also are voltage sensitive, but they open slowly. They open after a bit of a delay. And so the sodium channels close, the potassium channels open. Now potassium wants to flow out of the cell, right? So because there's a lot of potassium on the inside and not very much on the outside, potassium's positive charge. So now you've got a positively charged ion flowing out of the cell, and that brings the membrane potential back
down again,
which is the resting level. So that's the basic idea, but there's two important ideas that we need to sort of elaborate on. And the first is that when you hear this description, this is what almost everybody thinks. And this is what I thought when I first heard this description. I thought, okay, here's the way this works. The sodium channels open. So there's a lot of sodium on the outside. The sodium channels open and all the sodium flows in. Now you've got a lot of sodium on the inside. Then the sodium channels inactivate, the potassium channels open, and all the potassium flows out.
Comes out. Yeah.
Now you've got a lot of potassium on the outside. So now all these gradients have been reversed, and then you've got these pumps that pump everything back the way they were before. And that's not how it happens. That's not the way it works, right? The amount of sodium that flows in a single action potential hardly changes the concentrations of sodium at all.
Yeah, there's so much sodium on the outside.
It's a tiny, tiny amount. And the amount of potassium that flows out hardly changes the concentration of potassium at all. It changes it just a little bit, right? And of course, it does change because sodium flows in and potassium flows out, but it's a very small change. When the neuron is at rest, the reason the inside is negative is not because the inside of the neuron is filled up with negative charges. It's because there's a tiny, 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 tiny imbalance of negative charges, a few extra negative charges on the inside. At the peak of the action potential, the reason that the inside is positive is that there's a tiny excess of positive charge. There's a few positive charges that aren't matched up with negative charges.
Okay. Yeah.
So the next problem is, why does the action potential move? Why does it propagate down the axon? And the answer to that is, it's actually very, in a sense, it's very simple. It has to do with the voltage-gated sodium channel. So let me explain. Okay. Now, at the initial segment, there's an action potential. So when the action potential happens at the initial segment, the inside of the cell is like plus 30, but it's minus 70 all down the length of the rest of the axon, right? Now, positive charges and negative charges attract each other. So the positive charge that flows in at the initial segment, it gets attracted down the axon. It starts being pulled down the axon because it's negative down the rest of the axon. So the action potential starts, just because of this property, because the whole axon is covered with sodium channels, the action potential just keeps propagating down the length of the axon. It's never going to stop, right? In fact, if you could have an axon that stretched from here to the moon, and the action potential
just keep going. Okay.
So one metaphor that I use is, if you've ever seen, like in these old movies, I don't think they do it anymore, like in old Western movies, the guys would light like a fuse that was attached to like sticks of dynamite, right? And so the flame is going to burn down the fuse. It starts at one end of the fuse, and then this bright flame moves down the fuse. Well, why is that? It's happening because the whole fuse is flammable. And so you light that one end, and that one end bursts into flame. That's like the initial segment. And that gets the adjacent region of the fuse hot enough that it bursts into flame, right? By analogy, it's the same with the action potential.
Those potentials, yeah. And when you say regions of the axon, is that split via myelin?
So that's the basic mechanism for propagating the action potential, right? And if it's an unmyelinated axon, that happens continuously. If it's an unmyelinated axon, there are sodium channels all down the entire length of the axon, right? And so this process just is continuously regenerated all down the length of the axon. If it's a myelinated axon, it makes the action potential propagate faster. So one way that you can make an axon propagate faster is to make the axon fatter. The larger the diameter of the axon, the faster it's going to propagate action potentials. So evolution has had to come up with a way of making the axons as small as possible, as thin as possible, while at the same time making them propagate action potentials rapidly. And the way that the mammalian or the vertebrate nervous system does that is to wrap the axon up in myelin. Myelin is an insulation, but it's interrupted periodically by places where the myelin isn't there, right? And so in your
there's the gaps.
Those are called nodes of Ranvier, right? So they're periodic, they're about a millimeter apart, periodic gaps in the myelin. The idea in a myelinated axon is that this whole process that I've described with the voltage-gated sodium channels, that is only happening at the nodes. So what that means is that the action potential begins at the initial segment in just the ways that I've described. But instead of activating the immediately adjacent region of axon, that electrical signal, it actually spreads. It spreads farther. So instead of just having to spread to the immediately adjacent region of axon, it's actually able to spread farther and faster. It can spread all the way down to the next node. So it turns out that process, that mechanism, works better or faster than having the whole axon covered with sodium channels. So think of the difference between a runner, like a sprinter, and a speed skater, right? So a sprinter, they're constantly having to move their feet to get themselves going forward continuously, right? Whereas what a speed skater can do is a speed skater can push off and then they can glide. So they're starting to slow down to the point where maybe they need a little more oomph. They push again and then they glide again, and they go and then they gradually start to slow down and then they push off again. And so the runner, clearly the speed skater is going to go way faster than the runner.
Exactly. Yeah.
The speed skater is like a myelinated axon, and the runner is like an unmyelinated axon. So in an unmyelinated axon, this process of sodium channels opening up and sodium flowing in is happening continuously all down the length of the axon. Whereas in a myelinated axon, like in a speed skater, the action potential gets a boost.
Yeah, and then glides another one. And yeah.
And then it coasts to the next node and it gets another boost and it coasts, and that goes a lot faster. Right.
Yeah. Well, thank you for that great description, Dr. Ragsdale. Really appreciate you coming out and helping us.
No problem. It was my pleasure.
Now we're going to be looking at how exactly action potential can be measured. Abbey, would you mind giving us a little description of how that is done?
So it's pretty impressive that we can measure these tiny electrical signals in our brains, especially since they're so brief, like Dr. Ragsdale touched on. Scientists have studied the flow of the action potential for over a century, but only in the past few decades have we really been able to produce high-grade tools that give us high spatial and temporal resolution. One way to measure electrical currents in cells is with the patch clamp technique. So to carry this out, a glass micropipette is filled with an electrolyte solution and a recording electrode connected to an amplifier. So this solution conducts ions, which, as you know, hopefully by now, are really big players in the neuron's change in voltage. They're basically the ones that do everything. So there's also another electrode that's placed in a bath surrounding the cell as a reference ground electrode. This solution in the pipette could match the cytoplasm for a whole cell recording, or it could have a different ion concentration depending on exactly what type of recording you want to do. In a whole cell recording, the pipette is tightly sealed to the membrane to prevent leakage and to get a precise recording of the current. This can be done on neurons in cell culture models, dissociated neurons, neurons that are in brain slices, and in awake animals or animals that are under anesthesia. This technique has revealed a lot about the properties of excitable cells, such as neurons, and it's a great experimental tool because it reveals specific functions or channels in real time, depending on the manipulations you do to the cell, which could be something like introducing drugs. There are also some optical techniques like fluorescent dyes. Indirect dyes measure the change of ion concentrations during an action potential. So this might be like the influx of calcium or sodium, which would depolarize the cell. And that change would be detected and you could see it visually with the fluorescent dye. There are also direct dyes that are bound to membranes of the cell, and these dyes respond directly to the change in membrane potential through different mechanisms depending on the properties of the dye you're using.
All right. Thank you very much, Abbey, for that description. So action potential, very complex, but hopefully we've helped just a little bit. And in case this is still confusing you, just remember, unless you want to be a neurophysiologist, and if so, good luck, you only need to know this for your Psych 211 class. So stay in there. So, and hopefully you've come away with a better understanding of the physiology behind this process. And that concludes our second episode of the podcast. So thank you very much for listening, guys. And yeah, see you in the next one.